So we're going to first look at a ligand-gated ionotropic receptor. This is also a type of a plasma receptor. And so in the form that we see right here, this is closed. And so what happens is we're going to have some kind of signal come to the closed receptor, and this is on the extracellular side. And once the signal binds to the receptor itself, it's going to cause a conformational change, allowing the receptor to be open. So once the receptor is open, this is going to allow the transfer of ions. So like sodium can pass, or calcium, or chloride. And these two ions can basically act on this voltage-gated transport, voltage-gated channel, and this is going to allow more calcium to come in. And so the function of this specific receptor is that after the membrane potential of the potential by gated ions have been able to open up and pass through, the cell becomes excitable. This is going to alter the calcium concentration because we're now open the voltage-gated channels. And by doing so, we've increased the probability of an action potential, for instance, in a muscle or a neuron. And we know when we see voltage-gated calcium channels, we think of, our sarcoplasmic reticulum and stuff like that. Now, if we go into another area, which is our second type of receptor, we're looking at the G-protein coupled receptors. And so our G-protein coupled receptors are a little bit different. They're going to be a little bit more involved. So, looking at the G-protein coupled receptor, what it is a transmembrane receptor. And so this thing I'm drawing right here is actually a transmembrane receptor that's going to come through. And so we have our lipid bilayer, which is still here. And we also have an amplifier, which is used, I'm going to just title it as an amp. And this is going to be used in initiating the processes, allowing for everything to happen. And so our G-protein coupled receptor has basically two forms. The inactive form, which I'm going to draw right here, we have the G-protein, and it's bound to GDP. When bound to GDP, what that means is there's no signal coming in to activate it. So if we were to have some signal come here, this would change into the receptor would signal that this GDP needs to be phosphorylated by a kinase. And so let's kind of go into depth what exactly the G protein looks like. So our G protein is made up of three subunits: an alpha subunit, a gamma subunit, and also a beta subunit. And it is these three subunits that remain attracted to each other when GDP bound. So when we're GDP bound, what happens is the gamma and beta subunit are held tightly with the alpha subunit. Once we have a kinase come in and we have a phosphate come in to phosphorylate the GDP to GTP, we have something different happening. And the result of this is and I'm going to write this a little different. This actually becomes alpha, and it's now GTP bound. Once GTP binds to the alpha subunit, what happens is the gamma and beta subunit are going to end up dissociating. And it's one of those kind of arguments of affinity. If something has greater affinity, it's going to basically say, I'm going to hold you more. Can you let go of this guy? You can almost think of it as a guy with two different girlfriends. He's going to pick the one that he likes more. So in this case, GTP is the girlfriend he likes more. And now our GPCR, or a G protein couple receptor, is in the active form. And from here, these two subunits and the alpha GTP bound are going to interact with the amplifier protein. They're going to interact with effector proteins and have multiple different functions. And so in the next couple of videos, we're going to go over what these functions are, kind of break down what the different subunits do, because in our alpha group, we actually have more than one alpha subunit. So we have alpha I, which is an inhibitor. We have alpha Q. We have alpha S, and we also have alpha 12, 13. And these are all things that you're probably going to need to know for the exam. So we'll start getting into that in the next video. If you like the video, give it a thumbs up. Please subscribe to my channel.